Yeah, thanks for organizing this webinar series, and to the KC, of course, and thanks to your team for integrating our data as you have so rapidly, and that really was great to see at our annual meeting in March. Okay, we're really pleased to be presenting today, so hello to everyone who's in attendance or perhaps watching the recording. We are going to give a team presentation. There's 4 of us. And we're pleased to participate in this webinar. Thanks to the Knowledge Center and other CFDE leaders for inviting us to present this overview and highlights, as we're calling it. My name is Jeremy Yang. I'm a co-PI of the IDG Data Coordinating Center, DCC. My fellow co-PI is Christoph Lambert, and both of us are at the University of New Mexico in Albuquerque. Also presenting with me today are Anna Waller, scientist and project manager for the IDG Knowledge Management Center, KMC we call it, and the Resource Dissemination and Outreach Center, RDOC. So she did a lot for both. Jamie Holmes is also presenting in our team here. He has been project manager and developer of key resource Drug Central. And our 4th presenter is Tim Shields, project manager and developer of FAROS of NIH NCATS. Anna and Jamie and I are at the University of New Mexico and have been IDG contributors since its beginnings over a decade ago. Tim is at NCATS and joined IDG a few years in, taking over from Rajarshi Gua, not an easy task. Raja moved from NCATS to a senior position at Vertex Pharmaceuticals, so he's done very well. This webinar has 3 parts. First, some history and background, which I'll do. Then Anna Waller will present on the importance and impact of IDG over its lifespan. In part 2, I'll present an overview of the many IDG data resources. Then Jamie will present Drug Central, an exemplar and featured IDG knowledge base. In part 3, Tim will present FAROS, the IDG data portal. Given the time allowed today and how much is in IDG, this presentation will really just scratch the surface, but hopefully be a reasonable length so that you can so it doesn't drag on too long either. So hopefully we're being concise, but quite informative. But really, this is only going to begin to introduce the IDG program and its data resources, which we believe are of much relevance to CFDE and potential value for biomedical data science use cases, integrating common fund and other data sets, which is kind of our overall theme, of course. However, for those interested in learning more, we will provide suggestions, links, and references to an extensive corpus of documentation and publications. And please feel free to ask questions via the chat or chat via the chat amongst yourselves. But there's also the Q&A, as Mackenzie mentioned, for asking questions of us, the panelists and the presenters. And of course, you can ask questions after this webinar by contacting us, so please feel free. And this is the outline of the webinar, as I described. The IDG program was designed, as Mackenzie indicated, to improve our understanding of understudied proteins from the most commonly drug-targeted protein families. 1, G protein-coupled receptors, 2, protein kinases, and 3, ion channels. 2, catalyze research in understudied genes that have high potential to improve clinical therapeutics and human health. The NIH Common Fund sponsored IDG from 2014 to 2023, supporting a diverse array of experimental and computational research. Druggablegenome.net is the IDG website, and FAROS is the data portal, integrating many other data resources from IDG across NIH and beyond. Bottom left on this slide from the druggablegenome.net is a graphic which illustrates the extensive IDG network of institutions and collaborators spanning the US and including European collaborators, EMBL, EBI in particular, and also University of Copenhagen. Bottom right is from an IDG Toolfest event with a sampling of logos from IDG discovery tools and data resources. Like any successful effort, the key ingredients of IDG have been the dedicated and capable persons doing the work. This slide includes some of the PIs and leaders who brought to IDG a strikingly diverse array of expertise, spanning foundational basic sciences, such as, for example, kinase research from cancer biologist Gary Johnson of UNC, GPCR research from neuroscientist Brian Roth, and the computational informatics and data science capabilities of the Knowledge Management Center, Resource Dissemination and Outreach Center, CATE, MC, and RDoc, and the Cutting Edge Informatics Tools groups. Tudor Aprea was the PI or API of the KMC and RDoc, conceived and led many of the computational innovations, including the Target Development Level TDL classification system. And Tudor was at a UNM before retiring to emeritus status and moving on to a senior leadership position in industrial pharma, very much AI-based. Now, since this is an CFDE webinar, it is especially fitting that we recognize Avi Mayan, pictured here as well, who is PI of the CFDE Links, DCC, and DRC, and who was a PI for the IDG, and in whose group developed a great many of the IDG data resources featured in this presentation. The IDG program was focused on the human proteome, and more specifically, the three most common drug-targeted protein families, as mentioned, and their protein coding genes. However, two other key entity types were prioritized as central or essential to this mission. Diseases, which can broadly include conditions or phenotypic traits, and drugs, which broadly can include ligands or chemicals, generally small molecule chemicals. So three entities, sometimes we say the holy trinity of drug discovery, and the relationships among them, and annotations about them. So these are the kind of data and relationships which are represented by IDG and which have really made the efforts coherent across these diverse institutions and investigators. Genes and proteins not in the prioritized families were also studied, guided by understandings of complex multifactorial polygenic disease etiology, systems biology, and polypharmacology. That's a mouthful, but I think you probably get it. Reductionism is an important way to do science, but we are trying, I think, all of us to think in terms of systems biology and more complex understandings. So that very much informed IDG research. Despite the complexity of the challenge and related data, a simple categorization method was found to be highly useful and popular, the TDLs, already mentioned, target development levels. And that's what this slide is about, TDLs. And the KC has already integrated some IDG resources, and in particular, featuring target development levels with that understanding, and as we encouraged. TDLs categorize targets into 4 levels from heterogeneous evidence measures, with highest level T-CLIN from activities in Drug Central, which are approved drugs with known mechanism of action. The bottom right figure is from the the right figure on the right is from the 2018 landmark paper, IDG paper, with statistics mapping TDLs to protein families and an expanded view of T-CLIN. Throughout the IDG program, and partly as a result of IDG efforts, proteins have progressed from T-dark to T-Bio, to T-Chem, T-Chem, to T-Clin. This slide includes some IDG news, mostly new publications from 2023 and 2024 this year, although funding for IDG largely ended in 2023, not completely, but largely. New publications and other products continue to appear from no-cost extensions and other support. Efforts are underway to sustain and enhance key resources, so there is reason for optimism that IDG will continue for some time in some form.